Cervical cancer is a serious disease that starts in the cell of the cervix, the lower part of the uterus. It mainly caused by persistent infection with high-risk type of human papillomavirus, especially HPV type 16 and 18. Here, how it works. The virus infects cervical cells and can integrate into their DNA. This disrupts normal cell function, causing genomic instability and eventually cancer. Globally, cervical cancer affects over 600,000 women every year and causes around 340,000 deaths according to WHO. The highest burden is seen in sub-Saharan Africa and part of Asia. But the good news, it's preventable. Vaccination against HPV, regular screening like PAP and HPV tests, and early treatment of abnormal cells can drastically reduce the risk.
So how does it infect cells and drive cancer? HPV infects epithelial cells by entering through tiny abrasions in the skin or mucosa. It attaches to the surface receptor and enters the basal cell through endocytosis. Once inside, its DNA is delivered to the nucleus and uses the host machinery to replicate. High-risk types like HPV16 and HPV18 produce two key oncoproteins, E6 and E7. E6 promotes the degradation of p53, a protein responsible for triggering cell death in damaged cell. E7 inactivates retinoblastoma, RB protein, which normally controls cell division. This leads to uncontrolled cell growth, genomic instability, and eventually cancer, especially cervical cancer. These mechanisms make HPV a major cause of virus-driven cancers in humans.
Cervical cancer staging based on the 2018 figure system begins with stage 1, where the cancer is confined to the cervix. If it is microscopic, it is 1A. Tumors that are visible fall under 1B, ranging from less than 2 cm in 1B1 to over 4 cm in 1B3. Stage 2 means the cancer has grown beyond the uterus. If there is no spread to the parametrium, it is 2A. Involvement of the parametrium bumps it into the 2B. In stage 3, the tumor may reach the lower vagina, pelvic wall, or affect kidney function. Stage 3C specifically includes lymph node involvement. Finally, stage 4 involve the bladders, rectum, or distant organs like the lungs or liver. MRI and PET/CT imaging are vital for accurate staging and treatment planning.
Epigenetic change affect gene expression without altering the DNA sequence. In cervical cancer, high-risk HPV cause two major epigenetic disruptions. First, DNA methylation silences tumor suppressor genes. Genes like RASSF1A and CADM1 become hypomethylated, meaning methyl groups block their expression, so the cell lose important growth and invasion control. Second, microRNAs or M1 RNAs. These small RNAs regulate gene expression. In cervical cancer, M1R21 is upregulated, promoting cell survival, while M1R34A is downregulated, removing its tumor suppressive effect.
In cervical cancer, HPV oncoprotein E6 and E7 interfere with key cellular pathways that regulate normal cell functions. First, the PI3K AKT-mTOR pathway, which promotes cell survival and growth, is frequently activated by E6 and E7 oncoproteins, as reported by Zhang et al. 2015. These abnormal activations allow cancer cells to avoid apoptosis and continue dividing. Next, the WNT beta-catenin pathway becomes dysregulated. Normally, beta-catenin levels are tightly controlled, but in HPV-infected cells, it accumulates in the nucleus, triggering genes that promote uncontrolled proliferation. Finally, the NF-κB pathway, which regulates inflammatory response, is persistently activated due to HPV presence. This leads to chronic inflammations and tumor-promoting immune signal, further supporting cancer progression.
First, vaccine 9 present virus-like particles of the HPV-L1 protein to trigger neutralizing antibodies against 9 HPV types, achieving almost complete protection in HPV-naive individuals. Despite the WHO's goals to vaccinate 90% of girls by 2030, only about 27% are reached today. To improve access globally, single-dose vaccine schedules are now being used. For diagnosis, high-risk HPV DNA or RNA testing has replaced Pap smear as the primary screening tool. We use P16-INFO-A staining to confirm transforming HPV infections, and self-sampling kits are helping boost participation. Cutting-edge liquid biopsies can detect circulating HPV DNA to monitor disease, and AI-based image analysis is improving screening accuracy. Finally, therapy goes beyond surgery and chemotherapy. We now use targeted drugs like bevacizumab and antibiotic drug conjugated tisotumab vedotin. PARP inhibitors are being tested for tumors with DNA repair issues. Promising options like bispecific antibodies and HPV vaccines are also in development, aiming for longer-lasting remissions.